Okay. All right, so we did have a question on whether or not McGraw-Hill will be included in the final exam. There are no specific questions that only dealt with McGraw-Hill. So if we talked about them in lab, and they were also talked about in McGraw-Hill, then that's a really good resource for you to review. But no, I will not take specific questions out of McGraw-Hill. That also goes for hypothesis. So if there's any OpenStax reading specifically that we did not discuss in lab, right? So there are things that we discussed in lab that your reading helped solidify those different topics. And so I would definitely urge you to go back through hypothesis and look over that material. But I'm not going to pick out little details that we specifically have not talked about. There is not a hands-on lab portion for this, this exam. There is a question where you will need to go to a station, and it is, you go up, you wait for the person to look at what they need to look at, they sit down, you guys take turns. But there is no hands-on. I'm not going to make you cut anything. I'm not going to make you actually do like an onion squash or anything like that. You do not need to know about entropy. You do not need to know about law of thermodynamics or anything in that example. Okay. You will need to know about karyotypes, pedigrees, so I'll go over those. We will talk about lethal inheritance. We will talk about incomplete dominance, photosynthesis versus CR testing, so the CO2 indicator. Yeah, so we will talk about, I'm not going to ask you very specific photosynthesis questions, but there are some phases in photosynthesis that we did talk about. So Hill's reaction, Calvin cycle, we did talk heavily about those. And so I would definitely, not the very little details, but the overall big picture you should know. You will not need to know specifically about how to find absorbance or wavelength. But if I was to ask you a question on whether or not, when looking at absorbance, if your rate increased or decreased, should you have a positive slope, a negative slope? Should it be a very steep increase or not so much? So you should recognize the graph that you did for that one particular lab. That helps. And I think that was it for all of the questions that I got. Let me just quickly go through. Yes, there will be a question about non-disjunction. Know it for both meiosis 1 and meiosis 2. We need to know the types of blood that each type can receive and give. Not necessarily, but I would understand antigens versus antibodies. And if you had somebody that was given the wrong blood, what specifically would happen because of the antigens and antibodies within that person's blood, right? So you should know about agglutination and how that could react. Any other questions before we get started? So I'm going to put on the list here. So we're definitely going to talk about the last quiz because I have been grading them. So all of my Tuesdays are completely graded. I'm now working on section 16. So 16, that should be done within the hour after this. 16, 17, 19, that should all be done by the end of Monday. I am not coming to campus on Monday. I do have, my mother-in-law is having surgery, so I have to be home on Monday, but I do have my virtual office hours. And so I do urge anybody with questions, please, I haven't had a lot of people come to my office hours, my virtual ones. So please come to those, and any questions that you have, we can have a one-on-one discussion with. All right, so we're going to go over the quiz. We're going to go over non-disjunction, okay, karyotype, pedigree. And what I'm going to say about the pedigree is I'm actually, once this is all done, once our review is done, I'm actually going to make a separate video specifically for the pedigree review sheet. Just so that if somebody does have a question about pedigree and they don't want to skip through this recorded lecture, that we can have like a separate video for it. So I'm going to answer questions very briefly about pedigree, but I am making a separate video for that review sheet. And that'll also give us some more time to talk about everything else that we need to talk about. All right, I'm good looking through, make sure that I haven't missed anything. So we have karyotyping on there, pedigrees. Was there anything else that you would like me to talk about? So we have the quiz, non-disjunction, karyotype, and pedigree. Please write in the chat, blood typing. Yep. Okay. Yep. So I'm just going to put non-Mendel, and that has our lethal inheritance. That'll be incomplete dominance, and that'll be co-dominance. Okay. Chi-square dihybrid. Chi-square and gametogenesis. Okay. Anything else? Yep. So when I talk about gametogenesis, that I'm going to be including oogenesis and spermatogenesis. Okay. Why don't we go ahead and talk about the quiz? So I was really happy about the amount of people that got the first one right. So that was good. So the first one, I actually really enjoyed making this quiz. I have a black lab, most of you know that. And everything that I wrote here on the quiz is 100% true. This is how labs get their coat color. My dog Bear, I went through Embark to get him genetically tested. He had some family history, so we wanted to make sure that he wasn't a carrier for anything, but I can see his coat genes, which is very, very cool. All right, so for the first one, you decide to get a puppy and you're looking for different breeders and you want to make sure that your puppy has a black coat. So this one here was just a very simple monohybrid cross. And you looked at the male dog. So our male dog was big B, little b. And the mama dog was little b, little b. Okay. And we wanted to know if you had a black dog and a brown dog, what would be the percentage of the offspring, or the probability, I should say, of the offspring being black? So we will do, and it doesn't matter if you have the male on the top line and the female going down or vice versa. I'm just going to do this. Just know the symbols in case that's written there, and then you can fill it out accordingly. And then you just fill it out. So bring it down here. And so this ended up being 50% black coat. Okay. So that one was pretty easy. I didn't find so far, as I'm grading, a single person getting that one wrong. So kudos to you guys. Awesome. Good job. Anybody have any questions on monohybrid cross? Pretty simple. Okay. All right, so let's go into, this one was a little bit more tricky, and I saw a lot of variations. Lots of different ways that you guys answered this question. Not as much as the different ways you answered the chi-square question, and we will get to that. Okay, so for this one, this talks about a topic called epistasis. And I know that we did not specifically talk about epistasis in the lab. You may have talked about it in lecture, but I really explained it as far as what that little e, two little e's would represent, which is why I told you that if you had something that had little e, you would only get a yellow coat. And so we had a dad here. Okay. And the mom, and my dog actually is big B, big B, big E, little e. And so this is dad. And this is mom. And this talks about the MC1R gene, which you guys are actually going to learn a lot about next semester when we talk about the rock pocket mice. It goes in conjunction with some of the other genes that we're going to be talking about for specifically coat color. All right, so we have mom, which is this one here. You guys can see that okay. All right, so let's do our dihybrid cross. Yes, I will give you a table. You won't have to draw in your own table for the dihybrid. That'll save you some time. Okay, so this is where I saw a lot of variations. Some students just brought down the letters one at a time. Please don't do that. Some just took variations of the first and the second. Please don't do that either. So can anybody tell me, let's say that dad's here and then mom is here. How would we put dad's genes down? How would they go at the top? Anybody want to take a guess? Yeah, you have to FOIL it. Yep. And so I had somebody ask me in class, I know I have to FOIL it, but I don't remember what that is. FOIL stands for something. It's an acronym, right? So FOIL is first, outside, inside, last. So we're going to look at the first set of genes, which is big B, big E. First, outside. You guys all see that? Yeah. And I had a lot of people do this correctly as well, except maybe they forgot to do like the middle part or the ending part. And I'm going to explain my grading and how I graded you on that in a minute. Okay, so first, outside, inside. So little b, big E, and then last. Little b, little e. Everybody see that? All right, and so then if we go here, we're going to do the same thing. First, little b, big E, outside, little b, little e, inside. Now it's really important to finish this as well because those E's are so important. Little b, little e. All right. And so what I'm going to do now is I'm just going to fill it out. So give me a second. And as I fill it out, I want you guys to fill it out too. Don't worry about this one being a different color. I'm not quite sure why it's doing that. Okay. Maybe. Now, when you do this on an exam, you can write in your genotypes and your phenotypes. That might help you as you go through. And I did actually, the one that I'm looking at now, that person did that exact same thing and they got the right answer. So that does help out. There was a couple of people that forgot what little e does. And so they did get the wrong percentage. And so if for some reason you did everything right, but you got the wrong percentage, I only took half a point off. If you didn't put these in correctly, unfortunately, you did lose all of your points. And if you just got one of these wrong, I took a full quarter point. Okay, we're almost done here. Okay. I'm going to step to the side real quick so everybody can see this. Okay, so that is our dihybrid. Now, let's go through and let's get rid of all of our yellow puppies. How many yellow puppies would we have here? Or the percentage, right? So out of 16, how many, what possibility would it be to be yellow? That one. So that's 1, 2, 3, 4. Does everybody see why those are yellow? It has to do with the little e. Okay. That blocks color from going into the hair, so you get yellow puppies. Okay. Next. We want, and we know from our previous example that little b, so little b equals what? What color is little b? Brown. We almost got a brown lab. All right, so brown. All our brown puppies. Okay, so we had 1, 2, 3, 4, 5, 6. Possibility of 6 brown puppies. So what does that leave us with? Which I think was 37.5%. Yeah. I'll move out of the way. So these, hold on. This would be a big B. Does anybody have any questions on the dihybrid? Are you sure? Okay. I'm going to erase it if there are no questions. Okay. All right. Question number 3. With the puppy example above, explain how the 2 laws apply. Again, this was very mixed between students. Some students got it right off the bat. Some were kind of there, some mixed the 2 up, and then others were completely in left field. Law of independent assortment. What does that say? So I'm going to give you guys a second. Yes, Claire, that is worded perfectly. Genes are inherited independently of each other, which is why we had a dihybrid cross, right? So if you are getting a B or an E, they are inherited independently of each other, right? So if you have a big B, that does not automatically mean that you're going to get a big E. Or if you have a little b, it does not automatically mean that you're going to have a little e, right? So they are independent. They are not completely attached. What would violate this law? Something linked genes. Yep, specifically. So linked genes meaning if they're on the same chromosome, that would definitely happen. Anything else specifically? So sex linked. So specifically, if something was sex linked, this would violate the law of independent assortment. What about law of segregation? So I got a lot of answers for law of independent assortment that actually was law of segregation. So law of segregation, what is that? And if for some reason you flip-flop the two, the question was worth 2 points. I only took off 1 point because I saw that you knew it. You just reversed it. So I only took off half credit. All right, so law of segregation. What are we saying about that one? Yep. Each parent can only give one allele to its offspring. So if you have a gamete here, and this one's from dad, and you have a gamete here, this is from mom, and if this one has big B, little b, and little b, you are only getting one from dad, one from mom. So somebody had asked a question that if the puppy is getting little e, how is it considered following the law of independent assortment? So if the puppy is getting the law of independent assortment doesn't specifically tell you about the other gene, right? So if we go back here, I just want to draw this real quick. So if we have a puppy here and we have a puppy that is big B, little e, little e, okay? The inheritance pattern of the B does not dictate if the puppy gets an E or not. That is what the law of independent assortment is saying. So if they get a B or they get another type of B, it's not going to say, well, then if they get this B, they definitely will get one of the little e. If something is sex linked, it's saying that if you get a B, then you are male, right? So it's where that B is on the Y chromosome. So it's linked that way. This is not linked in that way. Does that make sense? Hopefully I answered your question. Okay. All right. Now let's do the chi-square. I will say you do need to practice chi-square. There was definitely more people who didn't get it than did. And there were definitely some very creative ways that you did the chi-square. So please practice. Let's go through it. Now remember, I am not giving you a table. You will need to know your tables. 1, 2, 3, 4, 5, 6, 7. 2, 3, 4, 5. Squeaky. Sorry, guys. All right. All right, so we have black puppies, brown puppies, and yellow puppies. Okay. We're just gonna do black pups, brown pup, and yellow pup. Okay, so this is our O, excuse me, our E, O minus E, which I'm going to tell you O minus E, that's D. Okay, then we have D squared, D squared divided by E, and the sum. Okay. All right. So for this one here, the ratio, oh, I'm sorry. So D stands for deviation. Let me write that. D equals deviation. Okay. All right. So we know that the ratio is a 9:3:4. And it actually is. It's a 9:3:4 ratio. Okay. And that's black to brown to yellow. There was a litter. And out of that litter of 10 puppies, which is actually quite a huge litter, 7 were black. So this is what we observed. 7 were black. Oh, sorry. Yes, 7 were black, 1 was brown, and 2 were yellow. Okay. All right. This is where a lot of people did not know what to do. How many can tell me, how do you find E? Be more specific. What's the first thing that you should find in any chi-square problem? What should you do with your observed? Yeah, find the total. And we know it was 10. And why is that number so important? What are we going to do with that 10? Yes, we are going to multiply them by the ratios. So a lot of people did, oh, expected. I'm just going to put 9, 3, and 4 in there. Don't do that. Other people did 9 16ths, 3 16ths, 4 16ths. Awesome. But then they multiplied by the wrong thing. They multiplied by the 7, the 1, and the 2. Don't do that either. So you want to do 9 16ths, 3 16ths, and 4 16ths, multiplying that by 10. So I'm going to go off of the person here that's in front of me because their math looks pretty awesome. I also had a lot of people round, and there was a lot of funky rounding going on. So if you are going to round, make sure that you are following rounding rules. I had somebody round 2.5 down to 2. So if you did that, I took off 1/4 point. Now let's say you had put into your E the 9 to 3 to 4. So that row was wrong, making the entire thing wrong. I didn't mark you off for the entire thing. I just marked you off for that one line, and I marked you off -1. Okay. If for some reason your math was wrong, you messed up, I have one person mess up over here, 1/4 point off. If any of these numbers look wrong, just let me know. Like I said, I'm just quickly copying off one of the students, but I did the math earlier and it looks fine. All right, so you should have gotten, once this was all said and done, a chi-square of 0.84. I had some people, depending on how they rounded, it was about 1. If you didn't get that, right, so then that would have been wrong. And our null hypothesis was that there was no difference between, or statistical difference, I should say, between your observed and expected values of expected coat color, right? So that inheritance of coat color. And there was a lot of different ways that people stated that. So specifically, somebody wrote here about inheritance pattern. That's fine. As long as it had observed and expected, and you talked about inheritance, and you talked about the problem, I was okay with that. And then whether or not it followed Mendelian law, if this number was below your critical value. Now, how many degrees of freedom do we have for this one? 2. Yep, because there are 3 categories. So that means that we have your degrees of freedom -1, which equals, or your, I shouldn't say your, not degrees of freedom, your number of groups -1, and that gives you your degrees of freedom. Excuse me. All right. Now, for some reason, if your math was wrong, let's say you got 10 for that, and you said, no, we're rejecting our null hypothesis. If you wrote the null hypothesis and then you wrote rejected, I only took off 1/2 point. If you just said it was rejected, but you didn't actually write what the null hypothesis was, you got a full point off of that. You are going to be given the same little chart that you have had for all of your quizzes, and that was on your worksheet. So it will have the 3 different probabilities and the 5 different degrees of freedom, and you will need to figure out which one you are using. Excuse me, I'm sorry, guys. I will tell you that it's going to be the 0.05 for probability. It always is, but you will need to know as far as degrees of freedom, how to figure that out. Okay. Chi-square, done. All right, I'm going to erase this. So that was your quiz. Excuse me, guys. Sorry. All right. What do we want to go over next? Non-disjunction. We okay with that? Or is there anything that anybody wants to talk about before that? We're okay? Okay. See no objections. All right. So with non-disjunction, you have the splitting of a cell, well, going from metaphase to anaphase. And if those kinetochore microtubules don't quite retract properly, or it doesn't connect onto the kinetochore region of your homologous chromosome or that sister chromatid, it won't pull apart, right? So you have meiosis 1, meiosis 2. If it happens in meiosis 1, it's not as good for the gametes as if it happens in meiosis 2. So I'm going to show you what happens if it happens in meiosis 1, and then I'll do a side-by-side if it happens in meiosis 2 so that you can kind of see the end result. So I'm just going to split my screen. No, you don't have to draw all of meiosis. Definitely not. You won't have time. I just want the overall picture. Okay. All right. So here we have meiosis 1. Remember that we're going from one cell to 2 cells to 4 cells. I'm going to draw the same over here. Also, this is very general, right? So you know that for oogenesis, we go from 1 to 1. For spermatogenesis, we do go from the 1 to the 4. So just to make things easier, let's talk about non-disjunction in a male. Okay, it's obviously the chance is going to be a little bit more different in a female. So let's not complicate things a little bit more. We're just going to talk about sperm. Okay. Okay. All right, so let's say we have non-disjunction in meiosis 1. And let's say that they line up here. Okay, everybody see that? I know that's super tiny. All right, so again, remember that this is where those microtubules, for some reason, it fails. One of them does not connect on very well to our homologous chromosome set. And so when we go here, we would then have that. Okay. And then those would line up again. let's make sure I got that one right. This one would be. All right, so with non-disjunction, you would have here. Trisomy. Get out of the way. So for non-disjunction in meiosis 1, you would have 50% trisomy, so one more chromosome than it should have, and then 50% monosomy, one less than it should have. Okay, let's do it for meiosis 2, and then you can kind of see the difference. The other thing I want to say, and I don't know if we have it on our list here, but this is really important. And this was going back to the previous quiz. I said this over and over and over and over. Crossing over, when does that happen? Because I still got some of the wrong answers. So when does crossing over happen? Put it in the chat while I'm drawing this out. Yeah, prophase 1, I'm seeing still a lot of people telling me that it's during metaphase, and that is not true. Okay, so crossing over happens in prophase 1. Okay, I'm just going to step to one side for a second here. %, normal. See how you have the 3 chromatids there? Chromatids being now chromosomes. And then you've got 25% trisomy, 25% monosomy. Can everybody see that okay? I'm going to completely move out of your way just so that you can kind of see a big. Does anybody have any questions about non-disjunction and how it is different in meiosis 1 and meiosis 2? Awesome. All right, so I had someone talk about a karyotype. What specifically? So karyotype is very general, very broad. What specific questions do we have about a karyotype? And I'll wait for that to come in, and we'll move on to the next one. But whatever specific questions you have for the karyotype, let me know, and then I can go into it specifically. But it's such a broad topic that I don't know where you're not sure on, okay? All right, as far as a pedigree, okay, so let's, hold on for 1 second, I just need to have my husband bring me my paracord, or need paracord. Okay, or I'll lose you guys. All right, pedigree. So if we have a square, and we have a circle, no. Thank you. All right. Give me a trait. Thank you. Someone give me a trait, write it in the chat or yell it out. There we go, much better. Okay. I like that. Sweaty palms. All right. If we're doing sweaty palms, I need genotypes. So we got our phenotype, sweaty palms. What's our genotype? Let's do something different than S. S. So S's and C's and W's, those that are very similar with capital and lowercase letters, it makes things very hard for me to grade on exams and really to see the difference. So let's pick a different one that has a different letter. J's. Okay. So let's do J. And we'll do, nope. JJ, that'll be mom. And dad is going to be little j, little j. All right. We know that mom, you know what? Mom is going to be, let's say this is a dominant trait. Sweaty palms is going to be a dominant trait. We know that it has to be dominant, right? Because it's going to be brought through, and I'm also telling you, right? So if I know we had a question about this with the pedigrees before on whether there's carriers or not. And I also, in the review, there was one about sneezing, right? So for this one specifically, you either have sweaty palms or you don't, right? And so the dad does not have sweaty palms. And let's say little Joe and little Steve, Bob, let's call mom, call her Greta. I don't even know how you spell Greta. And we'll call this one Sam. All right. So Sam here does not have sweaty palms, neither does Joe or Steve. When I was talking to somebody in the lab, think of it as whether you have a green stem or not, right? Greta here, she has that capital gene, and so she is passing that on to Bob, and Dad gave the little one here. So Bob has sweaty palms. Okay. So if you're shaded in, you have it. If you don't, then you are not shaded in. Okay. All I'm really asking for you guys to do on the exam is to fill in what you know for your genotypes. Okay. If you don't know, just put a dash. I will make a separate video for the pedigree examples, the 4 examples that I had on that worksheet as soon as we are done here. Okay. Anything more about the blood typing? I mean, the pedigrees. We're going to move on to blood typing. Okay. All right, so I'm just going to move back. We did have somebody respond about the karyotype. So recognizing the shape, the size, the centromere position. So we are going to have three different types of chromosomes. Make these a little bit. Okay. What's the first one called? If I was to describe that first chromosome. First off, what arm is this and what arm is this? Okay, top arm is P, bottom arm is Q, and the first one is metacentric. Awesome. Then what? What comes next? So you've got, this is your centromere. Smaller P arm. Very small P arm. So what comes next? Submetacentric. All right. And then we have, it's the last one. Waiting for you guys. Yes, thank you. Telecentric. I spelled it much better than I did. I can look up the spelling. Telecentric. Okay. There is a fourth acrocentric. It's only in mice. Don't worry about it. Just remember these. And it's the P arm that is getting shorter. And you do not need to memorize which chromosomes are which, but you do need to recognize. You will be given a karyotype. You will need to label them. You will need to know X and Y, where that falls on a karyotype. You will need to know male versus female. You will need to recognize if it is monosomy or trisomy. So one more or one less. All right. Anything else on karyotyping? I'm going to erase this. Yeah, so an example of male versus female, if you have all 23 chromosomes, right? So you're going through all 23 chromosomes. Down here, you have your sex chromosomes, and you should have X if it is female, or X. And then I think it's just one for this one, which is male. So the Y, obviously, it looks a little funky. It's tiny. It's a telecentric. That is a great question. Somebody asked if this recording will be available to everybody, and yes, it will. I will post it. First, I have to post it up onto YouTube, and then I will make that link available. It's too big of a file to post up on Brightspace. And yes, that is all you need to know for karyotyping. Besides, obviously, what non-disjunction is. Okay. All right, blood typing. Very, very general. Specifically, what do we want to know about blood typing? Do you want me to give you an example and you tell me the blood type? We could do that. You guys want me to do that? Everybody see that okay? Okay. What is the blood type? Okay, so we have one guess. Anybody else want to take a guess? A positive. So if we were to draw this, okay, so there's my little vial. I'm going to explain what these are in a second. Hold on. Okay, everybody see that? So the pink dot here is going to be the Rh protein. And the blue dot is going to be the A antigen. The Y going to be the B. The anti-B antibodies. Okay. So it should look like something like that. Okay. Make sure that's everything that you wanted me to talk about. So this one here would be positive because it has a protein. It's positive for the protein specifically on the actual blood cell. Does anybody have any questions about that? Yeah, I can do that real quick. So somebody wants me to do a quick monohybrid cross. Let's say, can you guys plant over here real quick? Okay. Can you give me 2 blood types that will cross so you guys can see a monohybrid cross? B positive and AB negative. Okay. So we have B positive and AB negative. When they do blood type, you can do it however you want. They usually do little I's. And so you would do IA, IB. Okay. And then as far as this one here, we would have and now you told me B positive. Are we a solid B or are we a B with an O carrier? You gave me both. Which one? So we BB or BO? That's okay. Now, as far as positive or negative is concerned, don't worry about doing that in the cross. We're just going to talk about blood type as far as without the positive or negative. I'm not going to make it that hard. Okay. All right. So for this one here, you wanted me to talk about codominance, right? So here for codominance, A is dominant, B is dominant, O is recessive. Okay, so O kind of hangs out in the background. You can't get type O blood unless you have an O from mom and an O from dad. So you can never have a type O child in this situation. Now for this one here, you're going to have a type AB, a type BB, which would just be type B blood. Type A, or type B. So this one here, you have a 50% chance of having type B blood, a 25% chance of type A blood, and a 25% chance of type AB blood. Yes, you can write the chart any way that you want, as long as you understand it and I can understand it. If you don't want to put the I's in there, that's fine. No big deal. Okay. Any other questions on that? Okay. So for lethal inheritance and incomplete dominance, let's quickly talk about that. Now, I'm really surprised that nobody wanted to talk about photosynthesis or respiration or anything like that. So let's make sure that you do go back over that. Don't forget about that. Okay. Oh, I erased it. I'm sorry. Let's do that again. Sorry, somebody had a real quick question. We'll just really quickly do it again. All right. So we had, all right, let's see if we A, B, B, O. All right, so this would be A, B, 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 A, O, and B, O. So these are coming like this. Does that make sense? Hopefully that helps. Okay. I'll leave that up there till the person responds. Okay, so okay. So the person is asking, how did we know that the type B blood had an O? You don't always know. That is something that you would need to know from your parents and your grandparents. So my children are all type A. And if I was to do out their cross, I'm O and my husband is A. I can't give an A. I only can give an O. So I gave an O to all of my children. My husband gave an A to all of our children. So they are going to be AO just because they know our blood type. If they didn't know our blood type, they might not know if they're an AA or an AO. Hopefully that helps. Okay, lethal inheritance. So let's say you have a plant that can't produce chloroplast. So let's say we have, we'll do TT. Okay. If you have a plant here, that's my plant. Okay, and they in the background have their carriers, right? And they have in the background this recessive trait that if you get a full little t, you can't trigger something where you can't produce chloroplast. So if you're talking about lethal inheritance, this would be a 1 to 2, because these are the same. To 1. Right, where you have your genotypic frequency is going to be your phenotypic frequency is going to be we'll just talk about genotypic frequency. We'll talk about incomplete dominance later. So just lethal genes. For this one here, you have one plant that could never carry on that trait. You would have 2 plants here, or possibility of 50% of your plants that have that hidden gene. And then you have 25% of your plants that would probability that would have that lethal gene. Okay. When you talk about this for incomplete dominance, so let's say this was color of their petals. Okay. Where this would be pink, this would be red, and this would be white. Okay. This is where you have your genotypic frequency equal your phenotypic frequency. I don't like saying that for lethal inheritance because you can't, it's not something that you can really visually see. It's more in the blood, and it makes it very confusing. So I don't like to use those lines for lethal inheritance, but for incomplete dominance, phenotypes match the genotypes. Okay. Any other questions on lethal inheritance or incomplete dominance? And if you can't read any of my handwriting, just let me know. This is incomplete dominance. So if it was co-dominant, you would have two that were actually dominant. Great question, though. Okay. Last one is gametogenesis. So you have oogenesis and you have spermatogenesis. Specific questions. So forever who wanted me to talk about that specifically, what did you want me to go over? Did you have any specific questions on that? And while that person is thinking of questions, if there is anything else that you would like me to go over, please let me know. Anything about photosynthesis, respiration? Okay, try to know what happens during. Okay, so I got two good questions. Yes, so for gametogenesis, so spermatogenesis and oogenesis, yes, you will need to know what is happening as far as you're going to go from a spermatogonium. So if we were to draw the same. Over here. And then I like have one at the top here. So going from here to here, you have mitosis. We're going to talk about spermatogenesis real quick. So from here to here, you have a spermatogonium. And it's going to turn into a primary spermatocyte. Okay. The primary is going to go to secondary spermatocyte. Then you're going to go to your spermatids. So these are all spermatids. Mature spermatids are sperm. So these are then also, then they turn into sperm, but they're still haploid, right? So spermatids, you're not going to have another set of meiosis. That they just they develop, right? They grow, they mature. As far as oogenesis, same thing. So you have, I'm just going to draw it like this, and then I'll erase what we don't need to know. Okay. So you have an oogonium, goes into an oocyte. This is your yeah, because your primary will come down here. And I don't remember the very top of that one. I'll have to look that up if that's going to be also called your oocyte. But you have your primary oocyte here. Now, with this one, you only have one. This here would not exist. This is called your polar body. That dissolves, okay? This here is your secondary oocyte. This would be your second polar body. And that dissolves. So you go from 1 and you end with 1. Okay, and then I've got yes. Yeah, so does the secondary oocyte mature into the ovum? Yes. At which point do each do the gametes become haploid? So I'm going to have you guys, I'm not going to go into specific details about that, but know that we have diploid here, diploid here, because you have to be diploid to start the whole process, because everything is going to double. And then these are going to be obviously haploid down here. Because haploid is half of the chromosomes, right? If you're going to be going through, if you remember up here, you have your homologous chromosomes, right? And as you go through here, you have now those are actually going to go into your sister chromatids, where then you only have your single chromosomes. So if I was to ask you this question, not going to be super detailed. I want you to know that by the end, it becomes haploid. I don't need to know specifically what spot in the middle. It needs to be haploid. Okay. Out the way. Sorry. Yeah, and you will definitely need to know how to recognize your drawings that you did in lab. So the pollen tetrad, right? So if we had a tetrad, or if it was the second meiotic division. So if we had a pollen, and I think it looked something like that. So that was your second meiotic division. And then we had your pollen tetrad, which looked like that. You also had a seminiferous tubule, and you had the cat ovary, which had your secondary oocyte, and you had the follicle, right, that went around it. So go through that. All right. Questions on this before I erase it? All right, so somebody had a question about a chloroplast. Where's my green? My green is gone. That's okay. We'll use blue. All right, so if I was to draw a chloroplast, okay. What are these? What is on the thylakoid? And the stacks are called the granum. What's on the thylakoids? Nope. So you have here, this is your stroma. So what's happening here? Just gonna pull up something real quick. Yep. There we go. Come on. Oh, I've lost my Zoom. Hold on, guys. I lost you. Yeah. So the thylakoids are using the energy from the sun. Specifically what? I know you know this begins with a P. Yep. So you have all these pigments that are on your thylakoid, right? They are capturing that light energy. You need that energy to then be transferred into the stroma. So what specifically is happening in the stroma? So I've got some arrows. What comes out of our light reaction? So what's going on here? And what does the CC stand for here? So I want you guys in the chat. What does the CC stand for? What would be here? What would be here? What's over here? So let's say this is A, B, and C. Good. Yeah, organic sugars, you could always say glucose. So if you talk specifically about CC, what does the CC stand for? Yes, thank you. So this is where we have our Calvin cycle, and on here we have our Hills reaction. This is also called the dark reaction, whereas this is called the light reaction. It's called the light reaction because you need specifically light. You do have energy that is being transferred from the Hills reaction over to the Calvin cycle that fuels the Calvin cycle. But it's called the dark reaction because it is not directly taking in light energy. Sorry. Does that make sense? I know we've been here a really long time. Does anybody have any questions about that? Hopefully, I'm going back through the chat to see if we have answered everybody. Okay. Is CO2 increasing or decreasing when you have photosynthesis? CO2. Sorry, guys. So CO2, yep. So CO2 is decreasing. If CO2 is decreasing, what is that doing to the pH? You see why we learned about pH in the beginning? It all kind of ties together, right? So yeah, pH is going to increase, which means that it is more basic. And if we had a little vial, all of our little algae in there, what color is that going to turn after being in the light for a long time? If CO2 decreases and it becomes basic. Perfect. Okay. Any other questions? Rack your brain, go through your notes. Anything else that you want to ask me? How many feel ready for tomorrow? Or not tomorrow, definitely not tomorrow. How many feel ready for Tuesday, Wednesday, Thursday, or Friday, whenever you have it? I don't see any other comments in the chat. So hopefully this review helped. Again, if there's any other questions that you have, feel free to kind of hang out. I'll hang out for a couple more minutes. I'm going to turn the recording off now in case anybody doesn't want to be on